treat, find the molecular mechanism, what happens? What happens when we treat with TNF alpha? TNF alpha is a cytokine which induce endothelial dysfunction, as we previously discussed in first slide, endothelial dysfunction. And in part B, then we implicated the same mechanism can be used for cyanidin-3 glucoside in protection against endothelial dysfunction caused by TNF alpha. So for this we used human umbilical vein endothelial cells extracted from human umbilical cord using collagenase digestion. All the experiments were carried in these cells. So you know this, we have previously explained, this glutathione sulfoxamide is an irreversible inhibitor of glutamate cysteine synthetase, an enzyme involved in glutathione production. So what we do is we just inhibit this glutathione production and create an oxidative stress in the cells. And this oxidative can create reactive oxygen species, create an oxidative stress. And simultaneously, we treat with TNF alpha, that is cytokine to induce endothelial dysfunction. So for 24 hours, BSO was treated 1 millimolar, and on the next day, we treated with TNF alpha. And after this, we followed endpoints for understanding the adaptive response. We used glyphosate expression for the cell viability. For measuring intracellular redox status, we used glutathione content assay by HPLC and reactive oxygen measurement by 2,7-dichloro-dihydrofluoreshil diacetate assay, and Western blot for measuring NRF2, F1 proteins, and real-time PCR for measuring studying HO and NQ1 cytoprotective gene. And the next part, to study endothelial dysfunction, we analyze NF-kappa B using electrophoretic mobility shift assay and real-time PCR for BCAM, ICAM, and E-selective. So now about the result. We understand that in our experimental condition, that is 1 millimolar BSO, we found that the BSO was able to reduce glutathione content, deplete total glutathione up to 95%. As you can see this BSO, that GSH and GSH, that is reducing oxidase form, is totally depleted, around 95% as compared to the control. And the TNF alpha was able to increase the oxidase form, means even the TNF alpha is making oxidative redox status in the altering the redox status in the cells, and BSO also changing the redox status in the cells. And when you see this result, that is reactive oxygen species, the BSO was able to produce reactive oxygen species twice than the control. And TNF alpha was around 0.6 times, but when we treat BSO and TNF alpha, the 2.4 more times of reactive oxygen was produced. So the BSO and TNF alpha is showing synergetic effect in producing the reactive oxygen species. Now, it's very interesting part here. You can see this cell viability for BSO, only less than 10% of cell death. We say that when we reduce the glutathione content, it's reducing 92% of glutathione content. It is producing reactive oxygen species twice than the control. But still the cell can survive. Why? This is a big question. Why? So we understand that some mechanism is protecting the cells from doing something. So we understand that if you see this TNF alpha treated cells, the viability was around 23%, and the cell death was about 25%, 25 to 30%. But when you treat with BSO, the cell death is improved, cell viability is improved. So BSO is helping the protection against TNF alpha. Why? What happens? Let's progress. What happens? So we say this NRF2 that brought with that stress pathway has been activated by BSO. You can see this result. The BSO activates NRF2. TNF was not able to activate this pathway. And BSO and TNF together, it's showing the pathway. Means BSO is protecting the cells from the effect of TNF alpha. That is endothelial dysfunction by activating NRF2. Further, we understood that BSO is able to increase NRF2. But how? We are proceeding step by steps. But how? So we studied this cycloheximate chase assay. This cycloheximate chase assay is a very specific assay which measures degradation of any protein or transcription factors in the cells, and it measures the protein synthesis of any transcription factor or any protein in the cells. Particularly, you know, this ubiquitination proteasome degradation in the cells is very important because it controls various cellular functions. Like, for example, you know this for NRF2, it degrades. NRF2 is a transcription factor in the cell, it degrades. It has a half time of 5 minutes to 3 hours. When it degrades very fast, so no activity, no protecting enzymes. When the degradation is prolonged, you have more activity and more of protecting enzymes produced, the protein synthesis. So we want to understand how the BSO was able to increase the NRF2 levels. So we followed this assay. So we used MG132. MG132 is just a proteasome degradation. It degrades it inhibits the degradation of NRF2. Proteasome, it inhibits the proteasome, which increases the NRF2 level more. So, and simultaneously, the NRF2 is synthesized in the cells. So it can be arrested. It can be arrested by CHX, cyclohexyamide. Cyclohexyamide is a particular universal inhibitor of protein synthesis, which can be inhibited, which can inhibit the protein synthesis. So what we did is, just to increase the NRF2 level at 0 time, we used MG132. Then we treated with CHX, so that we arrested the protein synthesis, so that we want to know no more NRF2 is available in the cells. And we treated with in another group CHX and BSO, means like we are arresting the protein synthesis, and we are like there is no more NRF2 produced in the cells. But we want to understand like what does the BSO is doing with the NRF2 degradation. So after this we did protein extraction and we performed NRF2 blotting. So in this result we can see this cells treated with CHX alone, that NRF2 is going down. Means the NRF2 is degrading very fast. But cells treated with BSO and CHX, it maintains a steady state. Means what? That this BSO is able to prolong the degradation of NRF2, and it is not involved in protein synthesis of NRF2. As reported in literature, that various NRF2 inducers are not involved in protein synthesis of NRF2. Now next we want to know that we understood that NRF2 is producing the BSO activating NRF2. Want to know that what happens to this cytoprotective genes like H1 and Q1. We found that at basal level, BSO was able to activate both this H1 and Q1, but TNA was not able to activate this pathway. As we previously referred that even this was not able to activate NRF2. So no more by even H1 and Q1. So this even with the BSO and TNF alpha is producing more H1 and Q1. It means that this BSO is able to protect the cells, activate this pathway NRF2 and H1 and Q1 pathway even with TNF alpha. Now we know this NF-propionic pathway. TNF alpha is a pro-inflammatory cytokine which activates NF-kappa B, and it produces various pro-inflammatory genes. Out of that adhesion molecules, which we are most interested. So it's a whenever you have reactive oxygen species in the NF-kappa is activated. But in our experimental condition, this BSO was not able to induce NF-kappa. Why? Because it's producing a reactive oxygen species, it's changing the glutathione content in the cells, it's like destroying the cells, BSO. But still, it's not able to activate NF-kappa, that is pro-inflammatory pathway, because this NF-kappa is inhibited by some other pathway, that is NRF2 pathway. So in this result, it proves that NF-kappa is inhibited by NRF2. So we are interlinking both these pathways. So this TNF-alpha is as usual, as by this chart, it produces NF-kappa, but BSO was not able to activate this NF-kappa. So, and further we wanted to know this NF-kappa induced BCAM and ICAM, that is the gene involved in endothelial function. We found that the TNF-alpha was able to increase this BCAM and deselected. These gene explanations are not good, they are bad guys. So this BSO was able to decrease this E-selecting and E-selecting BCAM. So we propose that this NRF2 pathway is inhibiting this BCAM and ICAM. So particularly we talked previously that NRF2 can activate by various pathways. One is ion modification at the KEAP one. Another one is kinases activation. There are various kinases activation takes place. It's a cascade. We have big family of kinases. But we particularly studied MAP kinases L1 and L2 because we found that these kinases are mostly involved in this NRF2 activation. So we found that BSO was able to activate these kinases, and as like we can correlate this NRF2 kinases activation, this NRF2 activating, then you have the cytoprotective gene. BSO but not able to, not by TNF-alpha, and BSO and TNF-alpha is activated. So we want to understand the importance of that. So we in NRF2 activation. So once we inhibit this NRF2, this F by PD9805, like pharmacological inhibitor, this NRF2 was no more able to translocate in the cells or no more able to produce the cytoprotective genes. So we come to know this F is very important kinases in activation of NRF2. Further to prove this, we have studied the cell viability. We can see this. Like previous result, it showed that BSO was able to protect the cell damage from TNF-alpha. But once we inhibit this L, the cells were no more, the BSO was no more able to protect the cell from TNF-alpha. Likewise, we studied this again cytoprotective gene. Once we inhibit this kinases, no more cytoprotective gene was able to induce by this BSO. You can see this graph. And in particularly, like we have seen this E-selectin was decreased by BSO previously, but once we inhibit this F kinases, this it increases the E-selectin. It means F kinases also is like NRF is activated by F kinases activated NRF2, then it activates this H1N1, which decreases the E-selectin. So first part, we conclude the first part that BSO reduces NF-kappa pathway by activating nuclear translocation of NRF2 and by producing the cytoprotective gene, that is ARE genes HO1 and N2O1. And this induced adaptive response can protect the cells from endothelial dysfunction or from atherosclerosis. Like NRF2 is protecting from atherosclerosis. So in the part 2, the same method was used. So we have designed one model to understand the function of NRF2 in endothelial dysfunction. In the part 2, we understand whether the same mechanism was used by binding 3-glucoside to protect the cells from endothelial dysfunction induced by TNF-alpha. So again, the same process was followed. The adaptive response, measuring all this NF-kappa and H1, all these genes. So we found that cyanidin was able to protect the cells from damage caused by TNF-alpha, like previously we did with BSO. So cyanidin was able to protect the cells at higher dose. Then again, the cyanidin was able to activate this NRF2 pathway with stress, without stress. With stress, without stress. This is very important because without stress, we know this all these molecules, cyanidins and other things, they have antioxidant activity. It can deplete, it can improve the production of SOD, sulfur superoxide dismutase, and increase in the glutathione content without any stress. So as we reported previously in our group, this NRF2 activation without any stress causes the increase of this glutathione and SOD. So again we measure this gene. Again the cyanidin is able to increase this gene, cytoprotective gene. Again we measured the ERK1 and ERK2. Cyanidin was able to activate this ERK1 and ERK2 in dose dependent manner without any stress. So all these results is correlated. NRF2 without stress, this cytoprotective gene is activated stress without stress. So ERK also without stress, stress. So we are correlating these things. Then even like previously, the cyanidin was able to decrease the NF-kappa B value. It means the cyanidin is activating NRF2 pathway, and this pathway is inhibiting NF-kappa B pathway in endothelial dysfunction. Then we measured again to further confirm whether this cyanidin is able to decrease this NF-kappa B induced endothelial dysfunction gene. We found that ICAM, BCAM, D-cyclin was able to decrease in dose dependent manner by cyanidin glucoside. And again we tested for inhibiting this ERK kinase pathway, whether this ERK kinase is also activated by cyanidin glucoside. We found that once we inhibit this ERK kinases activated by NRF2, no more protection from the cyanidin, and no more induction of NRF2 by cyanidin. So this is the classical pathway, NF-kappa B pathway that the TNF-alpha activates NF-kappa B, and NF-kappa B activates various addition molecule gene. And this another pathway, what we are talking like this NRF2 pathway, cytoprotective pathway. So we come in a conclusion that NRF2, NF-kappa B is inhibiting NRF2, NRF2 is inhibiting NF-kappa B. In the literature, there are so many work interrelated. We are mentioning that NRF and NF-kappa B, but nothing has been proved very clearly. So we say that NRF2 in our study, NRF2 is inhibiting NF-kappa B, as we see all this pathway later. Particularly with this result. If you see this result, with this NF-kappa B P65, it's a unit of NF-kappa B. NF-kappa B P65, it's a unit, like NF-kappa B has many units. There are 4 units. Out of this 1 unit, which is required for activation of endothelial adhesion molecule gene. So we found that the cyanidin protection from this decreasing of the cyanidin decreasing the NF-kappa B was no more able to decrease when we inhibit this ERK pathway. So we confirm that NF-kappa B is inhibited by NRF2 pathway with the activation of ERK kinases. So now the scenario is changed. Previously we used to say the antioxidant, it scavenges oxidant in the cells, and it puts an end to the production of oxidant. But now we say that the cells are no more involved in oxidant antioxidant binding, but it activates the cell signaling pathway to produce and protective enzymes in the cells. So might be there is one receptor, might be, we don't know. So our conclusion is that cyanidase is a good polyphenol, but it can be helpful in pathologic condition associated with inflammation and oxidative stress. The possible mechanism of cyanidin glucoside is by activating NRF2 to inhibit NF-kappa B pathway involved in endothelial dysfunction. And in this context, we say that all natural NRF2 and H1 inducer like C3G can be useful in protecting against vascular diseases, especially atherosclerosis. This is the end of this my research here in Messina. I have been gone to UK for 3 months from September to December 2011 in the Department of Nutrition Medical School, University of East England, Norwich, UK. So we screened collective flavonoid, like a lot of flavonoid screened for its anti-inflammatory and vascular activity. We selected 10 different combinations, and in that it was just 3 months, 4 months period. So I was just involved in method development, in developing the method for toxicity assay by WSTSA in UVEX and vascular activity screening, that is NADP1 oxidase inhibition assay in UVEX, and anti-inflammatory like flavonoids depleting TNF alpha in DHP1 cells. So I thank all these people, Professor Antonella Sanya, Dr. Samot, and Dr. Francesco Cimino, Twitter, and our group professors Domenico Bucambetta, Maria Teresa, and Antonio in UK, Pauline K. and